So very happy to open our panel discussion today around synthetic biology and really the new dimension that this brings us in translational medicine. So I'm Bob Deans, I'm CSO at Synthego, and I'll moderate the panel today. We're really fortunate to have Ali Nahvi with us from Spark Therapeutics, and who heads their new therapeutic medicines unit, and Tim Liu, the founder and CEO at Senti Biosciences. And so earlier this spring, you read likely the partnership that had been formed between Spark and Senti, and I thought we would have the panelists discuss that, both their personal background and corporate perspectives going into it, and then some back and forth on value of the partnership in our sector. So we'll go ahead and open up with Ali and move to Tim. They'll each take about 10 minutes to give an intro, and then we'll have time for dialogue. So, Ali.
Oh, thank you, Bob. It's great to be here. Maybe I'll just start by talking a little bit about Spark and transition into applications of synthetic biology, why we saw value in sort of exploring that space for advancement of gene therapy products. So some of you may know Spark is a fully integrated commercial company that is committed to discovering, developing, and delivering gene therapies to patients. And we have a focus currently on retinal diseases, as well as hemophilia, lysosomal storage diseases, and neurodegenerative disorders. Since 2019, we've been part of the Roche Group, and today we continue to push for innovation all across the gene therapy value chain. And that really means for us to be able to deliver gene therapies to patients that we currently cannot treat, and it also means being able to provide treatments for more complex disease and indications. And I think that's really the space where we feel if you had greater degree of precision that was incorporated into the therapies, that would really be enabling to be able to treat those more complex diseases. So this issue about precision medicine, I think this is an important topic for today's discussion. That's where we see a lot of value in bringing in synthetic biology applications that perhaps were developed for other applications in their first iteration, but now also applying them for in vivo gene therapies to give you that, again, control over where and when you are able to have your gene therapies be active. So this is, I guess, the beginning of how we began our conversations with Tim and the team at Senti. I think it's fair to say that in the initial stages, we really didn't know what a scope of a collaboration would look like. We wanted to understand from the Senti team where they saw opportunities to grow their company and apply the biological logic as well as the gene circuit technologies that they developed for ex vivo cell therapies and apply them now in the context of in vivo gene therapy. So we began to understand where there was the greatest overlap. Eventually wound up through multiple conversations being able to identify areas that we could work together. And the rest is history. So now we have an ongoing collaboration. I think there's good rapport between the teams, and I think part of that was driven by the fact that we really sort of had discussions that were ongoing to really define the scope, and there was a degree of trust that was built in each of those interactions. And I think that really set the stage us for a collaborative kickoff start, as opposed to just throw everybody in a room and expect the two groups to work effectively together. So I think that fortuitously worked in our advantage. The last thing that I just want to maybe highlight is why did we decide to move as far as synthetic biology approaches for AAV gene therapy now? And I think it's fair to say that's a multifactorial answer, but part of it has to do with the emergence of advancement in the field of synthetic biology and leaders such as Tim and companies such as Senti. But it's also due to the fact that there are adjacent technologies that are very much enabling to synthetic biology. So we're thinking about here in the context of being able to read DNA, so sequencing, as well as being able to write DNA. Now, never has it been more cost effective to be able to do gene synthesis. But also, very importantly, it's the ability to now analyze and understand DNA sequences. So here we're talking about computational biology and bioinformatics capabilities. So bringing all these factors together, they're all converging to set the stage such that now we're able to generate larger number of variants of DNA sequences and explore them for different contexts. And I think this is why now for us, is the right time to be able to think about incorporating the synthetic biology capabilities for the purpose of advancing in vivo gene therapy platforms. So maybe with that, I'll just stop and turn it over to Tim. And obviously, I'm very interested to also hear about the perspective from the Senti side in terms of how we began our initial discussions and eventually developed those into a collaboration.
Yeah, thanks a lot, Ali, and thanks, Bob, for the introduction. I think I couldn't have said it better than Ali in terms of why it makes sense today to really think about applying these sort of tools and technologies to the cell and gene therapy space. Just by brief background, I've been working in this area since the early 2000s when, frankly, at that point, I think most people probably thought these were little Lego parts and tools and sort of fun things that you read in nature, but not really something that would ever make it into a patient. And that's in part because of a couple bottlenecks. One is, as Ali eloquently pointed out, like the ability to read, write, and even understand the link between genotype and phenotype was still really challenging at that point. It was expensive. It took a long time to do this sort of work. Two is like our fundamental ability to even modify human cells was difficult. A lot of the initial work started off in bacterial systems because it was essentially easy to do at that point. And we've made a lot of progress in that field since then. And then thirdly, and this ties into one of the reasons why we're really excited to work with Spark, is really this ability clinically now to actually take these programs forward. The ability to actually practically dose, viruses and cells into people, and then, have that be safe, have there be a lot of interest in pursuing these sort of clinical experiments, and ultimately push these towards products, is something we didn't have, 20 years ago. So the field has come a long way since then. We started Senti back in 2016 with the idea that, we were so inspired and awed by the amount of great work being done in cell and gene therapies, showing that these first-generation products could really make an impact in patients. But with the idea that, if you think 5, 10, 15 years out, and even beyond then, to this whole new class of medicines that we're going to start using, there would definitely be need and a push to actually make these products more precision, more precise, more controllable, et cetera, because that's fundamentally what's possible and something that people would want. And ultimately, that comes down to the ability to deliver on one end, but also the number two, the ability to control and regulate. And so I think the partnership, as Ali mentioned, was really exciting. We were able to sit down and really talk through where we could best apply the technologies. One of the things to think about when you're trying to incorporate some of these next-gen control elements or gene circuits into any product of interest is, even though we have certain categories of things that we can build, logic gates, control circuits, et cetera, ultimately they should be customized and optimized to the actual delivery modality and the ultimate, patient application, right? Because if you're treating cancer versus autoimmune disease versus some rare disease, so the level of control you may need, where you're going to tune the thresholds, turn things on or off at, what cells you want to hit, they're all going to be different. And that's basically because the disease biology is there. So we have the ability to customize those. But the really productive and fun part of it is really sitting down with collaborators, sitting down with clinicians and hearing about patient needs, and really then figuring out, hey, we have this technology, we can now customize it to solve this big problem in the clinic. So I'm happy to talk more about sort of what we do, but and in this particular context, but also I think Bob may have some questions more broadly about how we think about these technologies being deployed in the overall industry. So thanks.
Yeah, I think I actually warned him. I have to ask him when he first understood he was a synthetic biologist. I'm curious about what his mother thought when he was 6 years old and what he was playing with that defined him that way.
Well, I started my undergrad as a computer science guy, but this is towards the time when the first draft human genome project came out and the first gene switch papers came out. So it was just amazing that you could do that sort of thing. Frankly, initially I was just enamored by the sort of the fun part of it, so that's why I jumped into the field. But I think we ultimately realized there was a ton of applications for this in the therapeutic side, and made my switch pretty soon there afterwards.
Yeah, that's great. Ali, I have a question for you around launching this internally within Spark, and how hard a sell was it to take this upstream, and what were the risk points that popped up?
So I can speak for Spark's research organization. I think we are very mindful of being, again, a bit more future-oriented. It's not really about where we want to be in just the next year. It's also about where we want Spark and the gene therapy field to be in the next 10 years, right? So now is the time, again, going back to that point, to start to think about bringing a greater degree of precision and control over gene therapies. Again, being mindful that this is going to take some time for these technologies to be incorporated and be evaluated for both safety and efficacy preclinically until we're really able to roll them out in a clinical setting. But we want to be engaged in that now because there are some distinct, I think, limitations associated with the current state of the platform. And I think as members of the cell and gene therapy field, it's really incumbent that we're address those limitations and try to unlock them such that ultimately we're able to deliver these therapies to a greater number of patients. So I don't, to answer your question, I don't think it was a very hard sell. At Spark, we're very mindful that there are going to be enabling technologies, again, across a spectrum of different fields that we feel will be able to be put together in a cell and gene therapy product, probably in a modular fashion that will eventually differentiate the product for both safety and efficacy.
Yeah, I like the thinking a lot. You obviously put thought into your entry indications and what they were de-risking and how they were opening a broader commercial market. And I think you have 3 therapeutic areas that you're developing. Could you give a little bit of thought about what you're de-risking in one or several of those?
Yeah, so I think what I would say here at a high level is that I think it goes back to a point that Tim made earlier. It's about sometimes with therapies what we have is good efficacy, but what we are missing is the selectivity, right? And when you dial in selectivity, oftentimes it comes at a cost of the efficacy. So here really, it's about having a good understanding of the disease biology, understanding what are your on-target cell types, what are your off-target cell types that you're trying to avoid, and then building a sort of a thinking around what your product profile needs to be, and then identifying opportunities and technologies that you can see enabling you to ultimately get there. And I think from the perspective of engineering, there are obviously 2 components to AAV gene therapy. There's what you use to deliver the payload, that's the capsid, and then there is what is inside of the capsid, which is the payload. So I don't think from my perspective, I view this as either or. I don't think any breakthrough is going to be the solution to it all. It has to be multi-component. There's a large amount of efforts being undertaken by various groups and companies as far as capsid engineering is concerned to be able to identify variants that have better profiles as far as tropism, potency, immune evasion. I think that's part of the equation, but equally important is what you put into the capsid, right? So having a transgene that you're able to either modulate as far as what the disease state is presenting, or be able to potentially modulate through the use of an exogenous small molecule to be able to give that precision control, I think is important. And this is, again, a very large area of overlap with what we see with Senti's gene circuit technology and smart medicines in general.
Yeah, it's great. It's really good. Sort of the corollary, Tim, I think it was an easy sell for you with your executive team to get the partnership approved. I'm curious about how much of the dialogue as you were going into this was around efficacy for the therapeutic target versus safety. And generally, as you're talking to other clients outside of the Spark relationship, are people approaching you more on the safety or the potency side?
Yeah, it's a good question. I think actually both of those are actually pretty intimately linked with each other. I really like the way Ali's thinking about it, which is really trying to go after some key problems where you sort of know the biology pretty well already, and there's some key feature that you're trying to adjust for by introducing sort of this extra layer of engineerability. So I'll give a few examples actually for our own internal pipeline, just as an example, and then maybe that'll be useful to sort of generally answer your question. At Senti, we're focused to building a pipeline of internal allogeneic CAR-NK programs, and very similar to in the gene therapy side, we were looking for problems where there are known targets, known ones that you can go after for unmet cancers, but there's a need for greater specificity, which ultimately translates to a wider therapeutic window, and that means you can treat the disease potentially more aggressively. So, I think there's again, in the context of cancer, one of the key challenges we see, which is, I guess, not too dissimilar perhaps from the gene therapy space, is it's hard sometimes to find enough precise targets. Many of the targets we might want to go for in the oncology setting are also expressed on healthy cells. And if you're using a single target approach with an ADC or an antibody type strategy, you're going to naturally hit some of those, on-target, off-target, on-target, off-tumor cell types. So by being able to introduce like an additional receptor, be able to do things like logic, where you can say, kill everything that expresses tumor antigen A, but don't touch the healthy cells, that sort of level of precision ultimately lets you protect the healthy cells more and go more aggressively against the tumor cells. So I think, we envision that there are similar needs in the gene therapy space. Again, if you can perhaps shield those off-target cells from the effects that you don't want, that perhaps allows you to treat, the on-target cells in a more aggressive fashion. And I think that same concept applies into other cell types, other delivery vehicles, et cetera. So that's how we generally think about the problem. Initially, trying to go after areas where the disease biology, the models, are pretty well known. And if we don't have that internal expertise where we're building that in the oncology space, I think finding partners like Spark, who obviously have that disease area expertise, have delivery vehicles, it makes a lot of sense for us moving forward.
Yeah, it's really good. Question for both of you. I'm not sure that there's been much regulatory guidance on safety issues around synthetic circuits. If there is, and you could advise or say anything, it'd be great. If not, that's fine, and we can move to the next question.
I'm not sure the FDA has put out necessarily specific guidance. Obviously, we all know that the FDA has guidance around genetically modified, things that we're all doing, and I think these are not necessarily any different than that, other than they're sort of increased in terms of components and perhaps some complexity that you're putting in. So we work a lot internally to make sure we're building quality assays, we can manufacture these products, et cetera, and that's obviously important for us going forward as we push these towards the clinic.
Yeah, and I think I would follow up on that point and just say, obviously, there's already a certain precedent for this. So, when we think about the DNA payload that goes into a number of gene therapy products, that's already optimized. So it's not necessarily the transgene sequence isn't necessarily the natural sequence that you see. There's codon optimization that goes there. The promoter enhancers that are used to drive expression are oftentimes combinations. Now, I think what we're talking about in the context of gene therapy, there's just another degree. It's essentially upped to another degree with a degree of complexity that you don't see currently. But I think the basic underlying principles are very much the same. We're now evaluating more complex and different combinations than the products that we have currently, as far as the DNA combinations are concerned.
Yeah, it's really exciting. Related to that, we could talk a little bit about some of the adjacent technologies that are likely in your development stream as you go to commercialization. And so I'm curious if you would point to some of those that are either high risk or in the basket, and just your perspective there.
Maybe I'll start by that, Tim. So yeah, again, we can bring up the topic of delivery. You can generate the best-in-class payload. So one that gives you a response to a particular disease state or precise control in certain cell types and not in other cell types. But if you can't deliver it efficiently and safely, I think you still have a problem. So for us, again, we view that as a multi-track approach in which we tend to work with partners and have internal efforts to innovate on optimizing the DNA sequence to, again, bring that greater degree of safety and efficacy, and then simultaneously be able to address how we deliver, right? So can we deliver to tissues that we can deliver to currently, develop more novel delivery modalities that are able to give you, again, something that you cannot currently accomplish with the current state of delivery technologies. And then ultimately, the hope is that both of these efforts will converge and advance approximately with the same time frame, such that you can bring them together and combine them to give you that extra differentiated product. But I will also say, if we're able to engineer a payload that is best in class and yet have to still use the current delivery modality, that's a very good win as well. So it's not, again, either or, it's both. And we view this in a very modular fashion.
Yeah, maybe I can comment on three, I think, enabling areas that I think are exciting. One is the role of computation in all this. As we now can generate large, gigantic libraries of DNA sequences and really start mapping out genotype to phenotype, human design obviously is still important in the process, but having a more automated way to ingest all that data and actually sort of develop algorithms that help you sort of suggest actually new designs that you may not have come up with yourself as a human designer. I think that's really powerful given sort of the state of data generation and also the state of computational and machine learning approaches. Two is, perhaps one of the challenges that's always on the mind is manufacturing. These sequences, one of the nice things about it, at least in the AV context, we believe we can really make these sort of artificial sensors or promoters that we're building quite compact. But in other settings, let's say for our NK cells, we're actually putting multiple features in at the same time. And so be able to deliver those and get them into cells efficiently, there's a lot of process development and custom manufacturing know-how. That is important as we develop there. And ultimately, I think that's an asset if we can figure out not just how to design this stuff, but also engineer it. And then third, I think, is just more speculative looking forward. I do think that with the ability to design more precise therapies, ones that can go after multiple targets or have that precision or control built into it. The role of diagnostics and sort of precision medicine in terms of guiding which patient populations we're using, or how do we monitor responses and then sort of adapt the protocol for that particular patient depending on what's going on. It's an area that I think will certainly come up because now we're going to have the ability to more precisely regulate what these products are doing once they've gone to patients, or design them a priority, only go after certain targets and exclude others. And so thinking about a lot of learnings from the oncology space as well as others in terms of designing sort of targeted diagnostics and fitting that in now into the cell and gene therapy paradigm, I think is a really exciting opportunity going forward.
Could I make one comment following up on Tim's point about the enabling technologies and computational biology? I think this is something that obviously everybody's aware of how important bioinformatics capabilities are in the context of cell and gene therapy. But I think one thing that perhaps is sometimes missing when we talk about synthetic biology, at times there seems to be a misconception. There's a definite discipline here. You're not randomly, sampling sequence space here. And I think when I was a student, we would always talk that if you were just doing a random assessment of sequence space, if you reached a piece of DNA that was, I think, about 125 nucleotides in length, and you were to generate every single variant of that 125, so 4 to the 125th power, you would come very close to exceeding the mass of the entire universe. So you can see this is not a feasible approach. And 125 nucleotides in the context of regulatory DNA sequences is, I would say, on the very short end. So you have to bring in discipline and use, again, bioinformatics approaches to identify, I would say, building blocks, and then rearrange them and combine them in different fashions, and then generate diversity pools, and then screen them effectively. And I think, again, now that we have the ability to generate large variants cheaply and sequence them, and then, again, understand what the outcomes are in an iterative fashion, this is the perfect storm in terms of now applying these principles to improving therapies.
Yeah, I totally agree with you there.
Yep.
Yep.
Yeah, really good. So we're going to take 2 or 3 minutes and stop, and I believe that'll keep us on time. So a final question I'm curious about is what training or exposure would you advise people, students, or people wanting to enter the synthetic biology space? And is there any other school than MIT that offers that?
The field is growing quite a lot. I think there's definitely many areas that are doing great research in this space all over the place. I think any biological engineering department has really picked up on this need. And I think, as Ali mentioned, I think it's really this mindset of design, build, test, learn that really is imbued in this. It doesn't really matter which discipline you come from. In my experience, I was a faculty previously at MIT. We had people who were biologists, computer scientists, mechanical engineers. It doesn't really matter that much. I mean, you can learn the molecular biology. But what you really need to be able to put into place is that mindset of using engineering design. It's not just random sampling. It's not just sampling a few data points, but it's really trying to build, when you design any system, try to make it high throughput enough that you can collect enough data and then apply models to try to understand what actually you saw and actually use that to guide the next round. I think that's the most important thing.
Yeah, I would, obviously, Tim is the expert on synthetic biology. I would say from my perspective, I'm keen on evaluating and implementing technologies that wind up being enabling. It's just to be agnostic, right? So this is not about having a single toolbox and trying to fit everything into that toolbox. It's having a toolbox, that's your core strength, and really building that out, incorporating additional capabilities. And I think the agnostic outlook is really going to be something that we are going to be more comfortable with as these emerging technologies come to the field and become more and more validated, because obviously human nature, the default state is to be skeptical. And I think skepticism is obviously fine, but we still have to be open-minded in terms of, even though we view this enabling technologies as taking time to be validated, it's still very much worth it, because I think the positive outcome that you get has a huge upside.
Yeah, it's really great. Let's give a round of applause to the panelists today.